So you'd think a cancer genetic test is, well, a cancer genetic test, right? Kind of simple, but it's not that easy. It turns out that when you're trying to read a tumor's genetic blueprint, some are way, way harder to crack than others. Some cancers, they just hide their secrets a lot better. So that brings us to the big question, right? Why is one cancer's DNA basically an open book, while another's is this incredibly cryptic puzzle? That's what we're diving into. Think of this as a genetic detective story. We're on the hunt for clues to figure out why solid tumors are just so darn hard to understand. Okay, so in our little investigation here, we've got two main characters, if you will. On one side, you have blood cancers. You might hear them called hematologic neoplasms. And on the other, you've got solid tumors, which are way more common. And let me tell you, when you look at their genetics, they are worlds apart. And this really lays it all out. For blood cancers, it's often, a single, really clear genetic mistake, a driver mutation. The genetic code is pretty straightforward, kind of like reading a book. But solid tumors, oh man, that's a whole different story. Their DNA is just a chaotic mess. It's complex, it's unstable, it's all over the place. It's not a book. It's more like someone took that book, ran it through a shredder, and now you have to tape it all knocked together. Okay, so why is that shredded manuscript so tough to read? Well, our detective story really begins with the first big problem any detective faces. You gotta have good evidence. Right? I mean, before you can even start analyzing anything, you need a clean sample. And with solid tumors, believe me, just getting that sample is half the battle. So here's the deal. To get a genetic fingerprint, you need to actually see the chromosomes, but you can only see them when a cell is dividing. So the process is you get a fresh tumor sample, you put it in a lab dish to grow, to culture it. You're just trying to get the cells to divide. But, and this is a huge but, you're just crossing your fingers and hoping that it's the cancer cells that decide to grow and not something else. And this is where it gets messy. The lab dish basically becomes a contaminated crime scene. You see, the normal healthy cells that were surrounding the tumor, these are called stromal cells, they're actually tougher in the lab. They grow like weeds. They can completely take over the whole dish, crowding out the cancer cells, and pretty much hiding all the genetic evidence you were looking for in the first place. And all of this work, all this effort, it's all just to catch the cells in this one tiny, fleeting moment. It's called metaphase. It's the only point in a cell's life when its chromosomes are all bundled up, condensed, and you can actually see them clearly under a microscope. It's the perfect genetic snapshot. Now, for blood cancer cells, getting this picture is pretty easy. But for solid tumors, oh, it's an absolute nightmare. Incredibly difficult. Okay, but let's just say our detectives get super lucky. They managed to get that perfect snapshot. You'd think they're in the clear, right? Nope. The puzzle is far, far from over. Because now they have to face the chaotic, shape-shifting nature of the tumor's DNA itself. It's like the suspect just keeps changing its disguise. First off, solid tumors have what's called high genomic instability. All that means is their DNA is constantly changing, just rearranging itself on the fly. Then there's karyotype heterogeneity. That's a mouthful, but it just means that different cells inside the very same tumor have different genetic screw-ups. Imagine trying to ID a suspect when everyone in the police lineup looks totally different. And to top it all off, that genetic photograph we talked about, the resolution is often terrible, so all the important details are just blurry. So let's recap. We've got a contaminated crime scene, which makes getting evidence hard. We've got a shape-shifting suspect that's impossible to pin down. But there's one last twist in our investigation, the human element. Because even when our genetic detectives get the evidence, they can't always agree on what it means. And this chart really drives the point home. This is based on 25 years of data from lab proficiency tests. Look at this. Labs are great at just spotting that something is wrong. About 95% of the time they can say, yep, there's a genetic problem here. But actually giving that problem the correct official scientific name? Wow. The agreement rate just plummets all the way down to 76%. Let me say that again, because it's pretty shocking. It means that nearly a quarter of the time, 24%, the experts couldn't agree on the right scientific name, the nomenclature, for what they were seeing. Think about that. 1 out of every 4 cases, they're looking at the same thing and calling it something different. That kind of disagreement is basically unheard of when they're looking at blood cancers. So what does one of these mistakes actually look like in the real world? Okay, here's a perfect example from a lab report. You see 46XX, that just means it's a female patient, and it shows a deletion on chromosome 1.
It looks totally legit, right? Super scientific. But there is one tiny, absolutely critical detail that's missing.
And here it is, the corrected version. See the difference? It's that tiny little 10 tacked on at the end. I mean, it looks so small, but those brackets and that number, they are everything. That little bit of text tells other scientists that this specific genetic problem was seen in 10 different cells they looked at.
So why is that little detail such a big deal? Well, without those brackets, another scientist looking at this report might think it's a constitutional problem, meaning something the person was born with. But with the brackets, they know it's something that developed just inside the tumor. And that is a huge difference when it comes to diagnosis and treatment. It's just incredible. In some of these tests, up to 20% of the lab experts completely forgot to add that one crucial bit of information.
All right, so we've been through it all. We've seen the struggle to get good evidence, the chaos of the shape-shifting suspect, and even the problems with how we interpret the clues. So that brings us to the final verdict, the big so what? Why does solving this crazy, complicated genetic puzzle matter so much? This table really wraps up our whole investigation nicely. Look at the two side by side. For blood cancers, everything's pretty straightforward. Getting that genetic snapshot, easy. The patterns, clear. The picture quality, good. Everyone agrees on what they see. But then you look at solid tumors, and it's literally the exact opposite on every single point. It's hard to get a sample, the genetics are a total mess, the images are blurry, and even the experts argue about the details. And this, right here, is the reason this all matters so much. Solid tumors aren't some rare thing. They're the vast majority of cancers people get. So figuring out how to read their chaotic genetic code isn't just some fun science project. It is the key. It's the key that's going to unlock the next generation of treatments, therapies that can target a tumor's unique weaknesses and create truly personal medicine that will save lives. Now, the challenges here are obviously immense. But, the pace of innovation is just incredible. So it leaves us with one final question to think about. As our genetic tools get better and better, which piece of this puzzle do you think we'll solve first? Are we going to master getting the sample? Or maybe we'll use AI to make sense of the chaos? Or will we just get better at agreeing on what we see? That is the next chapter in our genetic detective story.